So now we're going to talk about Hox genes, and what Hox genes are they're really important for development in organisms, and that's what we're going to talk about. So what a Hox gene is a transcription factor, and the reason it's a transcription factor is because it has a homeobox domain within the sequence, and another definition or another reason that it is a Hox locus or Hox gene is because it's located inside the Hox locus, which has a bunch of other LOX genes inside it.
So what's a homeobox domain?
A homeobox domain is a sequence of DNA inside the sequence for the Hox gene, and what it does is it causes the protein that's being transcribed to localize the DNA, and that's really, really important because one of the hallmarks of a Hox gene inside the Hox locus is that it inhibits things, or the more posterior Hox gene inhibits the more anterior Hox gene, and we'll get more into that later. So these are all the criteria that has to be met in order to have a Hox gene. Homeobox must be in the locus, posterior dominance, and then there's another thing called, there are paralogs of them, and that's really relevant or obvious inside mammals. Inside a mammal, there's 4 Hox locuses, Hox A through D, and each one has like a different, there's like 13 Hox genes inside it, and each one has like 1 or 2 missing. They're all kind of different from each other, but in the end, Hox 1A and Hox 1B are actually very, very similar to each other, and they're known as paralogs. So Hox 1A, Hox 1B, Hox 1C, Hox 1D are all paralogs of each other, and that goes down the line for all of them. So going back to posterior dominance, the ability for, or the property of posterior dominance leads to this thing called collinearity, and in an organism that's developing, these genes actually sync up to a location inside an organism. So if this is our organism, let's say it's like C. elegans or something. Actually no, because I don't know how C. elegans works, but say this is like a Drosophila embryo. Hox, and this is, okay, so if this is our embryo and these are our regions of expression for the genes, what's going to happen is Hox1 is going to start being expressed in this region throughout the entire organism, and then Hox2 is going to start being expressed in this region, but it's only going to start here, and then the same goes for Hox3, it's only going to be here, and then Hox4 is only going to be here. And that's actually really, really important because each one contributes to a different region or a different structure for the organism. And how that works is because of posterior dominance. What happens is Hox2 will inhibit the activity of Hox1, and it's going to do that in the entire region that Hox2 is being expressed. So it's going to inhibit it, the entire region here, and that will lead to only this region right here to be influenced by Hox1. So whatever Hox1 codes for, say for like antennas or something, antennas are only going to be developed here, and that's it. So then the same goes for Hox3. It's going to start inhibiting down here, down the entire way. So that means that only this region, or well, this region actually, will be from Hox2. And then the same goes for here, HOX3, and then the same goes here, HOX4. So then, and the reason that this works, the reason that HOX2 isn't being expressed here is because in this region of development, or this time and location of development, all of these genes are silenced. And only at this time and location does this gene get unsilenced. And the same goes further down the line. So further down the time and location, the genes on the locus get more and more unsilenced. So there are different mutations that can arise based off of these different silencing mechanisms. So there's two kind of classic mutations you can see. One is a duplication of a structure, and the other is an ectopic expression of one of the structures. So for duplication, that results in a loss of function mutation of or just a loss of function in general of an anterior gene / structure. And the way that works is, the way that works is if HOX3 gets knocked down somehow, it won't be able to inhibit HOX2 at this time and location. So if HOX3 isn't here, it won't be able to inhibit HOX2 until HOX4 comes into play, right? So these two regions will be developed from HOX2, or influenced by HOX2, until HOX4 comes and inhibits it. So that's how duplication works. And then, and then those mutations, or one of the mutation, or the things that we learned about, the mutation of, what the fuck is it? It's like the silencing factor or something. No, what is trithorax?
It's important in silencing.
Yeah. So we learned this in class. There's this protein, I guess. Yeah, that's what it is. It's a protein. Yeah. So trithorax is a protein, and it reduces silencing in the genome. And so if there's a mutation in that protein, it ends up being over-silenced. And that's really bad, because if you, at this time location, if this protein, or this HOX gene can't be unsilenced, then it's going to remain silenced. And that's how this doesn't get activated in time. So
HOX2 will produce again.
Huh?
HOX2 will produce again.
Yeah, exactly. It'll lead to the duplication. So, and then the other example is ectopic expression, and the phenotype that we talked about for that was the production of legs where antennas are supposed to be. And the way that works is a posterior HOX gene transforms an anterior structure. So something that's more posterior in the time and location is expressed at a more anterior time and location, which shouldn't work because these should all be silenced. And the way that works is this protein called polycomb, which promotes silencing, gets mutated and can no longer silence. So if at this time and location HOX3 is being expressed, then whatever HOX3 is supposed to induce is going to develop there. And that's when you see legs on the antennas.
Because the posterior is dominant to the anterior, so even though if HOX1 and HOX3 were both being expressed, HOX3 would still override it.
Exactly, yeah. Because if at this, say we have like these HOXs floating around, they're transcription factors, and they're just like hanging out, HOX3 is going to like
override them because of
preventing, because it's dominant,
and it's not silenced.
Exactly, and it's being expressed, and it shouldn't be, because it should be silenced, but it's not.
Okay.
So.
Sweet.